Good morning, everyone. Welcome to the BBS seminar series. My name is Dr. Shelley Thomas, and I would like to introduce our speaker. Dr. Maggie Panning Pearce is an associate professor here in the Department of Biological and Biomedical Sciences at Rowan University. Prior to joining the faculty at Rowan in 2023, Dr. Pearce earned her PhD in pharmacology from SUNY Upstate Medical University and completed her postdoctoral research at Stanford University. Dr. Pearce then began her independent academic career at the University of Sciences in Philadelphia in 2015. Currently, the Pearce Lab investigates the molecular mechanisms underlying neurodegenerative disorders such as Huntington's and Alzheimer's diseases. Her work aims to understand how toxic protein aggregates, a pathological feature of all neurodegenerative diseases, impact the survival and normal functioning of cells in the human brain. The Pearce Lab uses Drosophila melanogaster and mammalian cell culture experimental models to monitor the formation, spread, and toxicity of protein aggregates in neurons and glial cells. Dr. Pearce's talk today is titled "The Spreading Problem in Neurodegeneration: Insights from Fruit Fly Brains." Please give a warm welcome to Dr. Maggie Panning Pearce.
Thanks so much. So people are tending to sit in the back, so I'm going to try my best to project my voice, but please just tell me if you can't hear me. I want to thank the seminar committee for giving me this opportunity to share our lab's research with you today. And we're going to talk about several neurodegenerative diseases, but really with a focus on Huntington's disease. And this is, I just want to point out, art from Kathy Gibson, who was a caregiver to her sister Kelly, who developed Huntington's disease, who has since passed away. All right, so I don't think it will be any surprise to anyone in this room that our human population is aging at an unprecedented rate. Along with all of the benefits that come, with increased lifespan, is unfortunately typically a decrease in health span. Good. Okay. All right. So we see an increase in prevalence of many age-related disorders in the elderly population. You may be familiar. In fact, I'm sure everyone in this room has someone in their family who is suffering from one of these diseases. The most common, of course, is cardiovascular diseases. We have metabolic disorders, cancers, increased prevalence of all of these diseases, as well as neurodegenerative diseases, which are increasing at a very rapid rate in our population. In 25 years, it's estimated that the number of people who will be suffering from dementia worldwide, and dementia would include Alzheimer's disease, the most common form of dementia, but also some more rare and other Alzheimer's-related dementias, including the diseases that we study in my lab. So it's expected that the number, this of course is outdated by 5 years now, but the number of individuals suffering from these diseases will almost triple in just 25 years. And along with that is a global increase in healthcare costs that is expected to exceed $10 trillion. So this is going to be devastating for economies, governments, as well as the patients themselves and the caretakers who are taking care of them as these diseases progress. So increasing our understanding of the mechanisms that underlie these diseases is essential for the development of novel therapeutics that can actually modify the disease rather than simply alleviate symptoms, which is most of what these patients have available to them today. So symptoms of all neurodegenerative diseases are caused by defects both in neurons and glial cells. Of course, the diseases are named because of the loss of neurons, specifically in vulnerable brain regions that are specific for each disease. Defects in key cellular functions, so that includes gene expression, energy metabolism, as well as protein degradation pathways are primary contributors to the loss of neuron function and death. And as I said, the disease affects both neurons and glia really are first responders to neuron dysfunction and damage in all of these diseases. They activate immune-related signaling pathways that are thought to be initially neuroprotective, but over time develop into chronic inflammation that can actually be deleterious to the neurons surrounding them. So these glia may recognize dying neurons prematurely, which can lead to early synapse loss that is a distinguishing feature of all of these neurodegenerative diseases. And so learning about the balance between how neurons and glia communicate in the disease state as a result of dysfunction happening inside the neuron cell cytoplasm, but also the defects that are occurring to glial cells themselves will help us develop more targeted therapies for these diseases. So a common feature and really a primary pathological hallmark that's used in the diagnosis of these diseases, typically post-mortem, and I'm talking about all neurodegenerative diseases at this point, is the appearance of proteinaceous inclusions. These can be found either inside of neurons, or sometimes in the case of Alzheimer's disease, the primary pathological feature for many years was thought to be these A beta plaques, which are these huge inclusions found outside of neurons and glia. These inclusions contain misfolded and what become aggregated proteins and form because of a loss of protein homeostasis in the cell. So this is how cells deal with protein folding, as well as how they deal with clearance of misfolded proteins. So this deteriorates as we age, just because of normal aging, but also because of genetic mutations that are associated with these diseases. In some cases, environmental exposures and lifestyle can also contribute to the production of these aggregates, and you can see chronic traumatic encephalopathy, if you're familiar with the appearance of that in NFL players and others who have experienced TBIs, traumatic brain injuries in their life. So these protein aggregates are toxic to cells. They form in vulnerable brain regions and are associated with extensive loss of synapses in cells in those regions of the brain and are thought to contribute to the development of symptoms. Because of this, because they're toxic to neurons, they've been a major focus of drug development in the neurodegenerative disease field, although that's are. And unfortunately, recent FDA approvals of drugs targeting especially A beta plaques have been wrought with controversy. So we're still at a point where we don't have any disease-modifying therapies that are widespread for these patients. And so with the increase in prevalence that's expected, we really need to come up with better treatments for all of these patients. All right, so the proteins that I displayed on the previous slide share no sequence homology with one another, except that they each contain what are known as intrinsically disordered domains, which we shown up here, which cause partial unfolding and may be actually important for the protein's function, but can over time cause the protein to become aggregation prone. In some cases, it's a genetic mutation that changes a well-folded protein into a protein that misfolds rapidly. The process of aggregate formation follows what we call a nucleated conformational conversion process, whereby proteins that have difficulty folding, whether they start as a natively folded monomer or they are having a difficult time folding into their mature three-dimensional shape, will misfold and adopt this conformation that becomes predominated by beta sheets, which you can see here, which allows stacking of those proteins into a developing aggregate that achieves a lower free energy than the natively folded protein. So once this oligomer forms, and the size of that is still under debate for each disease, this is considered the primary nucleus. And so after this forms, we've reached the end of the lag phase of aggregation. And now we get a rapid exponential phase where there's the addition of new monomers into first protofibrils and then eventually mature fibrils. And these are what we see in the brain tissues that I showed, the images I showed you on the previous slide. It's thought that secondary, so what I just described is known as primary nucleation. Secondary nucleation events can involve fragmentation of these fibrils, whether it's at this stage or this stage, which can then accelerate the process. So you see the addition of a fibril seed of some size, it may be very small, it may be larger, can accelerate the process and lead to development of new fibrils. All right, so a lot of recent findings suggest that aggregate self-replication, as I just described it, occurs not only in an individual cell, which I show here, but can also propagate from cell to cell. So here we have a donor cell in which an amyloid aggregate is formed. So the specific type of beta sheet rich and soluble, very stable aggregate can form, and then either the entire aggregate itself or more likely a fragment, a piece that has broken off, and travel to a new cell, a naive cell, and rapidly accelerate the aggregation process in that cell. This type of behavior, which I'll tell you in a moment, is what we call prion-like behavior, has been shown for all of the proteins that form aggregates in all of these diseases here. So we have alpha-synuclein in Parkinson's disease, A-beta and tau in Alzheimer's disease, a protein called Huntington I'm going to tell you more about in Huntington's disease, and these two proteins here, SOD1 and TDP43, which are the primary pathological proteins in ALS. So the ability to spread between cells and seed new aggregates has been demonstrated for all of these proteins and resembles the behavior of prions, which are a very rare set of neurodegenerative disorders caused by the misfolding of a protein known as prion protein into a more stable, what we call amyloidogenic states, where it forms aggregates, as you can see here in a patient with a human prion disease, Creutzfeldt-Jakob. It was first described in sheep with Scrapie disease, which is why we call the misfolded form Scrapie, or SC for Scrapie. And if you're familiar with the mad cow epidemic from the 90s, bovine spongiform encephalopathy, or BSC, is also a prion disease. These diseases can be passed from organism to organism. That type of behavior for the aggregates I showed you up here has not been demonstrated, and so that's why we call them prion-like rather than true prions. This prion-like hypothesis for neurodegenerative diseases can explain the stereotypical patterns of pathology spread that we see in patients in postmortem analysis of patient brains with Parkinson's disease. So we're looking at alpha-synuclein spreading, which starts here in the brainstem and moves upwards into the cortex as the disease progresses. And for Alzheimer's disease, where tau seems to follow more closely the symptomatic progression of the disease, and you can see a very unique spreading pattern, although it is stereotypical within the disease set, suggesting that these aggregates are actually able to spread themselves. Might be a signal also involved, but the proteins themselves, everything points to them having the ability to physically move from cell to cell. And more importantly, these patterns of spread suggest movement along synaptic tracts in the brain. So those are major focuses of foci, of research into the mechanisms for how these might spread, and as well as drug targets. Speaking of, there's many events in the spreading process that could be targeted by therapies. So the questions that remain in the field are, how are these aggregates moving in the brain? And how does aggregate spreading contribute to disease progression? So we have multiple mechanisms. I'm going to quickly go through them here. So first would be the generation of the aggregates themselves. So the initial primary nucleation event, how is that occurring, and can we slow that down? There's movement between cells. So we have release from the donor cell, translocation between cells, whether that's across the synapse or between the non-synaptic region in the brain, entry into what we call the recipient cell, and that's where most of the research in my lab is focused. We have the replication event, so the propagation of that aggregate phenotype into the recipient cell, so the actual aggregation event occurring here. And then finally, we have more long-range movement. So if you think about synapses, those could be between very long regions of the brain that are very long distance from one another physically, but also the role of cell type vulnerability, as well as roles for non-neuronal and neuronal cells in this process. That could also involve regions outside the central nervous system. So as I said, all of these could be potential targets for therapeutic development. And there's a lot of focus on the events involving possible extracellular species in this process on being targets for antibody therapy. All right, so my lab is really focused on two diseases, and I'm going to tell you mostly about our work in Huntington's disease models. We study Huntington's disease because 100% of cases are caused by a genetic mutation, which makes it much more straightforward to model in the fruit flies and other experimental models that we're going to work with. So I'm going to tell you a little bit about Huntington's disease. So this disease was first described in 1872, or at least the most comprehensive description of it was provided by Dr. George Huntington, hence the name of the disease. It's a progressive neurodegenerative disorder that initiates with first some mild behavioral changes and loss of coordination that progresses into cognitive decline, and then involuntary movements is a hallmark of Huntington's disease. It used to be called Huntington's chorea, and prior to that it was known as St. Vitus's dance, and this is a depiction of that. Eventually, the patients lose the ability to walk, talk, and swallow, and usually that leads to their death. We know from postmortem analysis that the brain region most affected in Huntington's disease is the basal ganglia, and more specifically the striatum, which is involved in motor control, which helps explain some of the symptoms these patients experience. We see enlargement of the ventricles and loss of neurons in that region. This also tends to be where the aggregates associated with this disease accumulate. Finally, this disease predominates in families. I've already told you there's a genetic mutation that causes it. Before we knew that it appeared to have a dominant inheritance pattern. So for those of you in my intro to genetics class, we just talked about this. This should make you think dominantly inherited mutation. And in fact, that's what causes the disease. I just want to point out this is a local family. Donna here is looking at a picture of herself about 15 to 20 years prior. She passed away in 2010 from this disease. At the age of 68, the same year her daughter passed away from the same disease. So this is truly devastating for the families that experience this. So in 1993, the gene that causes Huntington's disease was identified by positional cloning. It was the first gene identified, disease-associated gene identified by this method, to be mapped to the short arm of chromosome 4, as shown here. It was previously known as interesting transcript 15. It was renamed Huntington for the name of the disease. So there's its location, and I'm going to show you the protein structure. It's a humongous gene, so over 3,000 amino acids in the primary protein structure, and I'm showing you here the sequence of only exon 1. So we have the protein amino acid sequence up here and the DNA sequence down here. There is a repeat of a CAG codon, which codes for the amino acid glutamine, which is abbreviated as Q here. If you inherit fewer than 36 repeats in this CAG tract, you will not develop Huntington's disease. People who do inherit Huntington's disease have more than 37, so that's our pathogenic threshold. And there's 100% disease penetrance above 40. There's also genetic anticipation in this disease, so parents can often pass on a Huntington gene that hits even more CAG repeats than they have themselves. So the disease, the protein that's produced by this gene can come in two flavors. So we have wild type Huntington with fewer than 36 repeats in that tract. So that encodes for a poly-Q tract here, polyglutamine tract, which is intrinsically disordered, but can be controlled by alpha helices at the N-terminal and C-terminal regions to it. However, if you have an expanded poly-Q region, as shown here, the intrinsically disordered region basically wins and causes the protein to accumulate into these amyloid beta sheet-rich fibrils, which I told you about earlier. So they resemble the same fibrils that we see in Parkinson's, ALS, Alzheimer's disease, and related dementias. All right, so because Huntington is a monogenic disease, this process here has been a major focus of drug development, and there is a big movement for what are called Huntington lower therapies. So these are drugs that target either the mRNA or the protein of mutant Huntington. Sometimes they can also target wild type Huntington, but the idea is that if we can reduce the levels of Huntington in cells, particularly the neurons that are affected by this disorder, we can reduce the appearance or the accumulation of these toxic aggregates and hopefully ameliorate the symptoms that the patients experience. All right, so if we express just the N-terminal exon 1, so a very small portion of the Huntington protein, we can recapitulate many of the features of Huntington's disease in both cells and animal models. And I'm showing you here cells. I'm going to tell you more about animal models when we talk about the flies. The first thing you can see is that the mutant Huntington protein will form aggregates. Here we have GFP, green fluorescent protein, tagged Huntington proteins, either with a Q150, well above that pathogenic threshold, or Q15 stretch, which is well below. So the wild type Huntington protein will appear diffuse and soluble throughout the cell cytoplasm and nucleus, whereas mutant Huntington will form these foci or puncta, indicating that it's aggregated. We can also measure aggregates by other biochemical and biophysical methods, including high resolution imaging by electron microscopy. And prion-like behavior for mutant Huntington has also been described. So if we treat cells that are expressing a wild type variant of Huntington that's tagged with an M-cherry fluorescent protein, which is shown here, so normally diffuse in the cell, and we treat it with unlabeled mutant Huntington aggregates, what we see is that there's a phenotypic change in the wild type Huntington protein in the cells. So this indicates that the aggregation phenotype was passed from the outside to the inside of the cell, which is a behavior you would expect of prion-like proteins. In addition, we see that there is a stronger correlation between loss of or defects in survival in animal models and the seeding capacity of Huntington rather than its accumulation into inclusions, suggesting that this seeding behavior could be more toxic than the actual formation of the late stage inclusions that we see in patients post-mortem. And in vivo imaging studies in patients have shown that the pathology seems to move between the basal ganglia here, where we see those medium spiny neurons that are most affected in this disease, and later in disease into cortical regions. So suggesting that there's a spreading mechanism also for Huntington's that we saw for Parkinson's and Alzheimer's. All right, so many mechanisms have been proposed for how mutant Huntington might propagate through the brain, and many of these resemble mechanisms that have been described for alpha-synuclein, tau, and other intracellular aggregates that I told you about earlier. So first of all, we can have passage of aggregates across synapses, which would help to explain why we see progression of pathology between synaptically connected striatal neurons and cortical neurons. We also see passage of aggregates through these membranous extensions. So some of these could be direct connections between cells called tunneling nanotubes, which is a cool name, describing these thin extrusions that connect to cell cytoplasms. Or they might be packaged into exosomes or what are called exophores that are released from cells. We can also have release or internalization of the aggregates via non-specific pathways or receptor-mediated pathways. And the focus of our lab is on the receptor-mediated pathways, which would be active transport into cells, as shown here or here. So about 15 years ago, as a brand new postdoc, and I was presented with the opportunity to develop a fruit fly model that would allow us to test these types of mechanisms for mutant Huntington in a fruit fly brain. So I turned to Drosophila and melanogaster, and I'm going to tell you why in the next slide. So why flies? So we decided to move to flies. There's some obvious advantages. They're small, they're simple. We can grow large quantities of them. They have a short life cycle. They're cheap to work with. They also have a short lifespan, so we can do aging studies in a very small time frame on the order of months and not years. They share a lot of gene conservation with humans. So about 75%, approximately 75% of the genes that are known to be implicated in human disease have a fly homolog. So we can study most human diseases in this invertebrate model. They're genetically tractable. So they've been used in research for over 100 years. We have a lot of tools available that allow us to express transgenes, which is what we do, with both spatial and temporal control. And they also have a real brain. So unlike other invertebrates like worms, I'm going to argue that flies, although it's very different from the human brain, do have a real intact brain. And the connectome of the fruit fly was solved and reported just last month. So this is the first brain in nature that's been solved by electron microscopy. We know exactly where every neuron sits, where every non-neuronal cell sits, where every synapse is formed. So we have all of the architecture and the anatomical knowledge about the fruit fly brain. Of course, the next feat is what is the functional interactions between these cells. So just for reference, 140,000 neurons with 54.5 million synapses in a tiny fly brain. All right, so most importantly for our studies, we have the ability to express transgenes in specific tissues at specific times during development. So we're going to use for all of our studies what are called binary expression systems, and I'm showing you one here. So in this system, which was first isolated from yeast and then introduced into fruit flies, we have the GAL4 transcriptional activator that is controlled by an enhancer that can be tissue specific. So in this case, I'm showing you a pan-neuronal enhancer controlling expression of GAL4. GAL4 will bind to this region of a transgene called the upstream activating sequence, or UAS, and will control expression of anything downstream. So in this case, I'm expressing a GFP tag wild type Huntington protein in neurons, and this is the expression pattern we expect to see. I'm showing you the image of the fruit fly brain here. This is all the synapses of the fruit fly brain rendered in 3D. Here we're looking at the mushroom body lobes, are these L-shaped structures. And then what are called the antennal lobes here in the central brain are shown here and here. So that's the part region of the brain we're focused on. Alternatively, if I express a mutant Huntington protein, in this case tagged with an M-cherry fluorescent protein, you can see that it's expressed in the same region of the brain, except that it looks very different. So the mutant Huntington protein is able to aggregate inside these blind neurons, which was important for our studies, and that's in contrast to the wild-type Huntington protein, which is diffuse. Now, if mutant Huntington behaves like a prion-like protein, it should be able to pass on that aggregation phenotype to a wild-type Huntington protein, as we saw previously in cell culture models, and indeed we saw that. So when we co-express in the same cells, these same neurons, the wild type and the mutant Huntington protein down here, you can see now wild type Huntington is 100% aggregated and overlaps with mutant Huntington. So we are able to see the aggregation of wild type Huntington proteins, but only so far when the two transgenes are expressed in the same cells. So really the question we wanted to ask was, can we see this between different cells? So we're going to use the same types of tools. We're going to express mutant Huntington in a class of presynaptic neurons known as olfactory receptor neurons. They have cell bodies in the antenna and the structure down here called the maxillary pulp, which sends odors and pheromones in the environment. So they're olfactory neurons, sensory neurons. They project axons into the central brain and form synapses in the structure called the antennal lobe, shown here in blue. Those synapses are formed with postsynaptic cells known as projection neurons, or PNs, in structures called glomeruli. So that's the circle here, and these are kind of raspberry-looking structures. Each of those kind of blobs in that antennal lobe is a glomerulus, where there's a lot of synapses, thousands of synapses being formed between these presynaptic ORNs and postsynaptic PNs. And so now we can use the GAL4, sorry, there's no laser pointer here. We can use the GAL4 UAS system to express wild-type Huntington only in the postsynaptic neurons, and a different binary expression system that's independent but analogous, where we use QF to drive expression of QUAS transgenes, in this case, a mutant Huntington protein tagged with M-cherry. So we can use red fluorescence and green fluorescence to detect these different proteins. They are originating in different cell types because of the genetic tools that we're using. So the prediction is if this mutant Huntington aggregate is able to move from a presynaptic to a postsynaptic neuron, we should see aggregated wild-type Huntington proteins, which would be evidence of seeding. And so we're looking specifically where these particular ORNs and PNs are synapsing. I'll just tell you that's called the DA1 glomerulus, which is shown here, which I've highlighted here in this image. So this is from one-day-old flies. We're not zoomed in enough yet to see anything interesting, so I'm going to zoom in on the next slide and tell you how we quantify these aggregates. So this was work from a team of a former graduate student and 2 undergrads who've now moved on to graduate school, medical school, who developed over a winter break this method of semi-automated quantification of these Huntington aggregates. So I'm going to hopefully play this video. So we collect these images by confocal microscopy, which gives us 3D images of this region of the brain. And what we're going to do is first segment the M-cherry channel. So this is our mutant Huntington aggregates. We're going to identify each of those puncta in the raw data set as objects. And then the next step that's going on in this video right now is filtering of the red aggregates that are also green. So those would be the seeded wild-type Huntington aggregates. We've restricted our data analysis to only green aggregates that are also red. So we're likely undercounting where the red aggregate might not be visible. But this would be our strongest evidence for seeded aggregation of wild-type Huntington across the synapse. And so using this method, what we did is we analyzed flies at different ages. So I'm showing you 1-day-old flies here with the raw data on the left for M-Cherry or GFP, and the segmented data on the right, indicating that these are the aggregates the computer software has identified. And you can see as the flies age, we start to pick up seeded wild-type Huntington aggregates in our green channel, and those increase over time. I'll show you the quantification by graph here. So there's a lot of bars here, but if you just focus on the trend, we see an increase over time in both formation of mutant Huntington aggregates as well as seeding of wild-type Huntington aggregates. If you look at the y-axis, the number of these seeded aggregates is much smaller than the total number of mutant Huntington aggregates that we have, and that's not surprising because these are supposed to be rare events that take times in humans, tens of years, decades to occur. All right, so this was really strong evidence that we're able to model prion-like transmission or propagation of aggregates associated with Huntington's disease in fruit fly brains. And so we next wanted to ask how. How is this happening? And this is where we can start to take advantage of a myriad of genetic tools available in fruit flies. So first we looked at neuronal activity because loss of synapses is a well-known feature of all neurodegenerative diseases. And before that, we see synaptic instability. So prior to loss of the synapses from the brain, there are likely to be defects in neuronal signaling, transsynaptic signaling. So we can use tools to either silence or overactivate neurons to see if that affects the transmission of these aggregates. And what we saw was a little bit surprising. So we used this genetic tool called Shibire-TS. Shibire is the Japanese word for seizure. So these flies that have a defect in this presynaptic protein are unable to, or have seizures. That's one of their phenotypes. And Shibire is the fly homologue of dynamine, which is a GTPase that's involved in endocytosis of synaptic vesicles from the presynaptic surface. So if you don't have active dynamine, you can't release neurotransmitters. So these are in effect silenced presynaptic neurons. So we've restricted Shibire-TS to our presynaptic neuron population. We raise the flies at what's known as the restricted temperature, which allows that temperature-sensitive mutant to block synaptic activity. And what we saw, if you look at the green bars here, is that actually increased transmission across the synapses. So when we have silenced neurons, which I would argue are dysfunctional synapses, we enhance transfer, which is perhaps not surprising if you think about what's occurring in the neurodegenerative disease brain. We next looked at cell death. So in this case, we were able to express this viral protein called P35, again in our presynaptic neuron population. And P35 is an inhibitor of caspases that are involved in cell death. So if we inhibit those caspases, we inhibit cell death. And the effect that we saw was that decreased transmission across the synapse. If we targeted P35 to our postsynaptic neuron population, we saw no effect. So cell death was specifically involved in release of aggregates from those presynaptic neurons and their transmission to postsynaptic neurons. All right, so the next question we asked was the role of non-neuronal cells in this process. So I've only briefly mentioned glial cells so far. I'm going to tell you more about them in a moment. The question is, do glia regulate mutant Huntington aggregate burden in neurons? This was the initial question. So we know that phagocytic glia that have the ability to phagocytose or clear debris from the brain are important for normal brain homeostasis as well as in neurodegenerative diseases. And we know that there are a lot of, I'm not expecting you to see all of these, there are a lot of genetic risk factors in pathways both in microglia and astrocytes in the mammalian brain that are important for clearance of neuronal debris, both in response to acute injury, so traumatic brain injury, for example, as well as in neurodegeneration. And so defects in this process can lead to the accumulation of debris in the brain, which of course can be toxic to both neurons and glia. And so we targeted a key phagocytic receptor known as Draper in glial cells. This is a well-known scavenger receptor. It has a homologue in mammalian cells. So this is a conserved pathway that we're looking at. Draper is known to recognize debris that's caused by acute injury to the fruit flies and lead to internalization, engulfment, and clearance of that debris. And so what we did, or what I did as a postdoc, was to injure our fruit flies by removing their antenna which severs the ORN axons where we're expressing mutant Huntington. That leads to degeneration of the remaining axon material. And you can see by 7 days post-injury, the majority of it is cleared. When we delete Draper, so using a Draper null allele called Draper delta 5, what we see is that blocks the clearance, which is expected, but it also increased the number of mutant Huntington aggregates, and if I show you the quantification, by about 2-fold. So we got twice the number of aggregates when Draper was deleted from the fruit fly, suggesting that not only is Draper involved in acute injury responses in the fruit fly brain, but it's also recognizing something about the neurons expressing mutant Huntington and causing it to engulf those and degrade them. So then we asked a question I never anticipated asking, which was whether mutant Huntington aggregates could transport or transfer between neurons and glial cells. So my thought originally was that glial cells would reduce Huntington burden in the brain and thereby slow transfer, but in fact had the opposite effect. So we do see transfer of aggregates from neurons to glia. So you can see the appearance of seeded wild-type Huntington aggregates here. We're now expressing our wild-type Huntington protein in the glial cytoplasm, not postsynaptic cytoplasm. And when I delete Draper, we completely block that. So Draper's not reducing the ability of these aggregates to transfer. In fact, it's enhancing it. So why is that? Is kind of the focus of the last 15 years of research in my lab. So the next question, could Draper-mediated aggregate engulfment contribute to spreading of Huntington across synapses? And we saw the same effect. So it's shown in a different way here, but if you look at the filled symbols here compared to the open symbols, which are the Draper knockouts, you can see that we reduce the ability of Huntington to transfer across synapses when we delete Draper. And just to remind you, Draper is a glial gene. It's not expressed on neurons in the fly brain. So this suggests that glia are directly, actively involved in transferring aggregates across synapses. And so this led to 2 models that we then tested. The first is that the aggregates might directly transfer across the synapse, and Draper in these glial cells is doing something to the synaptic environment to enable that. Or the aggregates transfer indirectly through the glial cytoplasm to the postsynaptic neuron. And Kirby, who is a PhD student in my lab, took on this tour de force experiment where she integrated 3 different binary expression systems. So now we're using GAL4-UAS in glial cells, the QF-QAS system in presynaptic neurons, and another system called the LEX-A-LEX-O system in postsynaptic neurons. So she needed to integrate 6 different transgenes into these flies, which is not easy to do. But she was able to do it, and we were able to get beautiful data showing that we saw aggregates that colocalized with Huntington proteins from ORNs and glia before we saw aggregates that appeared in PNs. So it seemed that these aggregates were moving in a circuitous route from presynaptic ORNs to glia and then to PNs. We also never saw the appearance of aggregates that only had signal from ORNs and PNs. They always had glial signal. So this ruled out this pathway, and instead suggested that this indirect pathway was what was responsible for aggregate transmission, mutant Huntington aggregate transmission across these synapses in the fly brain. All right, a closer look. So now we're even more magnified looking at these aggregates. So I'm showing you the raw data with the segmented data for our mutant Huntington aggregates, raw and segmented for wild type Huntington aggregates. And if you look at the merged images here, what you may notice, these arrows are pointing to, is that these wild-type Huntington aggregates are indeed seeded by mutant Huntington. We see the presence of mutant Huntington, but that the average size of those mutant Huntington aggregates is much smaller than the whole population. And I'll convince you by looking at thousands of aggregates that the ones that are able to seed, so these are mutant Huntington aggregate sizes, the seeding capable aggregates in the darker bars compared to the entire population in the lighter bars, the mean size is much smaller, suggesting that there is an inverse correlation between aggregate size and seeding competency. And we're not the only ones to have reported that. So another group did this in both a fly model as well as a cell culture model, and saw that the seeding activity seemed to predominate in smaller aggregates versus larger aggregates. Again, fragmentation causing secondary nucleation could be responsible for this. And then I just want to highlight work from a recent master's student in the lab who's looking at a mammalian model of this. So everything we've done so far, I've told you about, has been done in fruit flies, and we really want to expand the evolutionary conservation of our work. So we're looking at astrocytes. So this is an astrocytic, mouse astrocytic cell line and a human neuronal cell line. The neuronal cells are expressing mutant Huntington, tagged with M cherry. The astrocytic cells are expressing nothing, but we're going to stain them with an antibody known as GFAP, anti-GFAP, which labels only the astrocytic cytoskeleton, which is shown here in green. So anything that's green is an astrocyte. Anything that's red is a neuronal cell, or originated in a neuronal cell. And what Ryan was able to see is that some of these red aggregates, again, originating from neuronal cells, were completely internalized by the astrocytic cells. So this is a cell culture model where we just mix the two cell populations. We're able to see transmission of aggregates from neurons to astrocytic cells. And what he did further was to knock down the Draper homolog in these cells, which is known as MEGF10. So he used siRNA knockdown. We got depletion by more than 90% of MEGF10 levels in these KT5 mouse astrocytic cells. And he saw that that decreased transmission of the aggregates between the cells. So we have been able to develop a cell culture model to test the same hypotheses that we're looking at in the fruit flies. All right, so conclusions for part one of this is that mutant Huntington aggregates spread across synapses in the fruit fly brain via, and that's important here, via Draper positive phagocytic glia. So we never see any evidence of direct transport. It's all through glial cells. We saw that reduced synaptic activity and cell death of the presynaptic neurons promoted transmission across these synapses. And then there was a correlation between smaller seeding aggregates and the ability to transmit, suggesting that maybe there's an upper size limit for this transfer, or that aggregates are being fragmented in the process. All right, so this suggests that phagocytic glia are playing dual roles, or what I'm arguing are double-edged roles here in neurodegeneration. And we're not the first people to suggest this. In an Alzheimer's mouse model, there's a pathway that's mediated by the complement cascade here that can aberrantly and prematurely label synapses in mouse brains, expressing A beta, which is the aggregate that forms in Alzheimer's disease. That seems to be the same pathway that's turned on in the developing brain to remove supernumerary synapses during development. So extra synapses that are formed as a result of development. So this is a normal process, and it's thought that this is an abnormal reactivation of that same pathway in the neurodegenerative brain. And we know more recently that these complement cascade components, here we're looking at C1q, which you can see here is one of the factors that's directing microglia to these synapses to engulf them, labels the Huntington's disease mouse brain as well. And we see a decrease in synaptic, an increase in synaptic clearance in these Huntington's disease mouse brains as well, suggesting that the same pathway that's activated in Alzheimer's, leading to premature synapse loss, is also activated in Huntington's. So furthermore, the phagocytic pathway and endocytic pathway analogously is known to be involved or known to be defective in these neurodegenerative diseases. And there are a number of genes, some of which are genetic risk factors for these diseases, that are known to play a role in defects in endolysosomal pathways. And so the question we're asking is, what is the relationship between dysfunction of these phagocytic glia and neurotoxicity that's caused by mutant Huntington, and more specifically, spreading, the ability of mutant Huntington to spread? So Graham, who just graduated earlier this year, I guess almost a year ago now, in January, went after this question. So what he did is he expressed either wild type Huntington, and I'll show you in a moment, mutant Huntington, in the same ORNs that we've expressed in before, and he co-expressed with a membrane targeted GFP that we could use to track clearance of that neuronal debris following injury. So the same injury where we remove the antenna, we see clearance of the debris. When he did that in the presence of mutant Huntington, which you can see aggregated here, what we saw is that there was increased levels of GFP after injury, suggesting that these injured axons are persisting in the brain And we saw that at multiple ages, at multiple time points past injury. You can see the dotted lines all tend to be higher levels than the solid lines, indicating that there's a slowing of the clearance of this debris following injury. So suggesting that mutant Huntington is leading to defective glial clearance. Went after this by another mechanism by using transcriptional analysis. So we're using qPCR here. This is going to be very data heavy. I'm going to try and point out just the important parts, but what he was doing is expressing either GFP as a control, wild type Huntington GFP or mutant Huntington in all lower ends, minus plus injury, and then measuring levels of genes involved in the immune signaling pathway regulated by TOL6, which is the homologue of TLR6, and Draper-dependent phagocytosis. And what I'll point out here by the green bars is that mutant Huntington expression by itself, without injury, was able to upregulate 3 of these genes. So the receptors TOL6 and Draper, as well as the NF-kappa B homologue known as relish in fruit flies. And then all 6 saw defective increases in levels of all of these transcripts. So all of these genes are injury responsive. And the effect of mutant Huntington expression was to reduce that. So there's a reduced ability of these glia to be responsive to injury as a result of mutant Huntington accumulation. So he next went to the other side of the pathway. So we're looking at the kind of initial steps very early on following acute injury or Huntington aggregate expression. And we're going to look at the end stage, which is lysosomal degradation. So there's 5 known defects in lysosomes in neurodegenerative diseases. That includes a larger number, accumulation, enlargement. They're accumulating more debris. Over time, they lose the ability to acidify the lumen, which is important for degradation of the material in the lumen. The membranes can become ruptured, and that can lead to leak of luminal contents. So all of these have been seen in all neurodegenerative diseases, and we wanted to see if we could see the same thing, except this time, we're going to express mutant Huntington in neurons and look at lysosomes in glia, which had not been done before. So I'm going to quickly go through this data and tell you that levels of lysosomes increased, whether we used GFP tagged at the C-terminus or N-terminus of this lysosomal protein LAMP1. So we saw accumulation and enlargement, which I'm not showing you, of lysosomes in glia. I guess this is the enlargement. So you can see the darker red bars indicate that there's an enlargement, increased volume of the lysosomes that are associated with mutant Huntington. And then I'm going to show you using this lectin called the lectin-3, and we have data for another lectin called the lectin-8, which labels sites of lysosome or endomembrane damage in cells. So this will tell us our endocytic vesicles, phagocytic vesicles, and lysosomes rupturing in these cells. And we saw an increase in that in the presence of mutant Huntington, suggesting that neuronal mutant Huntington is causing defects in lysosomes and glia. And then finally, we decided to do for a genetic screen. So this is another advantage of using an invertebrate model such as fruit flies, is we can grow many fruit flies, we can perform many processes at once. And so what we did is we targeted these GTPases known as RABs, which cycle between GTP-bound active states and GDP-bound inactive states. And these enzymes are involved in organizing internal membranes in cells that are involved in vesicle trafficking. There are 33 RABs in the fly genome, all of which have a homologue in humans. There's 66 RABs in humans, so about half of those have a homologue. And so what we did, a former technician in my lab, Akram, who's now a PhD student at Drexel, he targeted every single one of those RABs individually and looked for the ability of mutant Huntington to transfer from presynaptic neurons to glia. So we knocked down these RABs specifically in glia and looked for effects. We used our 3 different controls, negative controls, and then we have Draper knockdown as a positive control. So again, that reduces transfer from neurons to glia. It also causes accumulation of mutant Huntington aggregates, as we saw previously. So after he knocked down all 33, these are the ones that didn't cause lethality. 3 of them did cause lethality. What we saw is there was 1. So we were lucky we got 1. And it's this gene called RAB10 here, and I guess the data are here. So you can see that RAB10 phenocopies the ability of Draper knockdown to reduce Huntington seeding, suggesting, at least initially, that RAB10 is involved in the same pathway as Draper. So RAB10 is a relatively understudied RAB. which is exciting, but also a little frustrating because there wasn't a lot of information about it out in the literature. But what I can tell you is that it's been described to be involved in almost every event of endocytic or phagocytic pathways. It's involved in phagocytosis of bacteria by macrophages. It hasn't really been described in glial cells, so that's the novelty of our work. But it has been described to release A beta aggregates from neuronal cells. There is a SNP in the RAB10 3' UTR, so not the coding region of RAB10, but the SNP is thought to be involved in resilience to late onset Alzheimer's disease. And you can see the pedigree here. These resilient individuals all tended to have this SNP, suggesting that RAB10, whatever that SNP is doing to RAB10 activities involved in resilience to Alzheimer's disease. What's even more exciting is that RAB10 is a phosphosubstrate, so it's a protein that's phosphorylated by this enzyme called LRK2, leucine-rich repeat kinase 2, which is a genetic risk factor in Parkinson's disease. So we've identified RAB10 in a Huntington's disease model. It's been reported to be involved, or at least be a risk factor in Alzheimer's disease, and now it plays a role in Parkinson's disease pathogenesis. So we're trying to tie these different neurodegenerative disorders together. RAB10 is phosphorylated in response to lysosome damage as well. So that was exciting for obvious reasons. It's been reported to cause the extension of lysosome membranes into tubules, and that could lead to release of lysosome content. So perhaps that could underlie the transmission of these aggregates from neurons to glia and glia to postsynaptic PNs. And it also, phosphorylated RAB10 also labels tau aggregates in mouse model brains of Alzheimer's disease. So everything points to RAB10 playing an important role in all neurodegenerative diseases, and we want to characterize it further in our Huntington's disease model. I'm going to go quickly through this because I know I'm running out of time, and there's a university assembled meeting at 3 that I know some people want to get to. We did show that RAB10 and Draper genetically interact. So when we have a null copy, one null copy of each in heterozygosity, we see a reduction in seeding of wild-type Huntington's, suggesting again that these two proteins interact genetically. RAB10 is an injury-responsive gene. We see transcript levels increase in response to injury to these flying neurons. And finally, we see association between mutant Huntington aggregates originating in neurons and different RAB proteins expressed in the glia. So here I'm showing you raw data and segmented data up here in the insets for RAB7. And you can see that there's a mutant Huntington aggregate here that's completely enclosed by a RAB7 vesicle, suggesting that's actually inside what's called a late phagosome in the endocytic process. We saw an increase in association of mutant Huntington with RAB10 and RAB7, but we actually saw the opposite effect for RAB5. And I didn't tell you about these different RABs, but RAB5 is involved in early stages of endocytosis. RAB7 is involved in late stages. So we think that RAB10 is behaving more closely to RAB7. It's involved perhaps in late phagosome or lysosome stages. And we have some compelling images to suggest that RAB10 membranes, so positive membranes, are surrounding some of these, or at least starting to surround some of these mutant Huntington aggregates here and here. All right, so for my second set of conclusions, I showed you that neuronal mutant Huntington aggregates can attenuate responses of phagocytic glia to acute injury in the brain. We've identified RAB10 as a novel modifier of prion-like transmission of mutant Huntington from neurons to glia. I've shown you that neuronal mutant Huntington causes defects in lysosomes as well as late phagosomes labeled by RAB7, and we think RAB10 as well. On the other hand, it leads to decreased abundance of early phagosomes, and I didn't show you data, but for what are called nascent phagosomes that form just after the internalization event. And so what we think this is pointing to is what we're calling a traffic jam model, where we have accumulation of aggregates at later stages in the phagocytic pathway. And perhaps the cells, because we see decreased internalization and formation of nascent phagosomes, perhaps the cells have a compensatory mechanism that is reducing engulfment and transit through this pathway. You can think of it like ramp signals on a congested highway, reducing the input, the flux of new material in, so the cell might be able to better deal with what's already in the system. All right, so what are the current and future directions that we're going after? So I have to apologize. I don't know if any of our undergrads are here. I didn't have enough time to get their pictures, so they may or may not be happy with how they're being represented as humanized fruit flies here. But, so we have a team that's led by a PhD student, Lily, that's interested in looking at what are the signals that allow glial cells to recognize neurons containing aggregates that lead to engulfment, as well as what is the role of autophagy, something I didn't tell you about, but another degradative pathway in glial cells in this whole process in transmission. We have another team led by Dawn, looking at what is the role of RAD10, this new modifier we've identified, in phagocytosis by itself, and also what is the role of RAD10 phosphorylation, perhaps by LARC, the homologue of LARC2 in fruit flies. What is the role of that in Huntington aggregate neurotoxicity? And then finally, we have a team of undergrads mostly that are looking at, can we develop a model for tau propagation that resembles what we're looking at for Huntington and kind of expand the impact of what we're doing into Alzheimer's and other related tauopathy models. All right, so I want to thank everyone who helped develop all of the data or collect all the data and analyze it that I've showed you today. So we have 2 PhD students who've since moved on to their next steps. So Kirby and Graham, a former technician, Abram, Ryan just graduated in May. And so Dawn and Lily are running the lab right now. And then a whole team of former undergrads who helped contribute to all the data that I showed you today. We have many collaborators who've shared numerous tools, especially in the fly community. I want to highlight, not in the fly community, but the Manners Lab. So Melissa Manners, right next door to us, who's providing us with primary astrocytes to look at whether what we're seeing in fruit flies is also true in mammalian cells. And of course, all the funding agencies that have helped support our research. I was asked, I'll do this in 1 minute. I was asked to talk about my career path. So very quickly, when I was a young kid, I wanted to be a doctor. You know, a lot of people probably have this story, all the way until I was probably a junior in college. I was headed to medical school, got into medical school, and then decided after 2 summers and during the school year of working in a research lab that I just absolutely loved research. This is what I wanted to do eventually. Even if I went and got an MD, I would have done this. So I decided to go get my PhD instead. So as an undergrad, I was studying spatiotemporal patterns of DNA replication in different cell types. This is an image from mouse embryonic stem cells. So I was doing a lot of imaging, live imaging, fixed cell imaging, looking at patterns, how they might differ at different stages of development. For my PhD, I stayed at the same university, but I joined a different lab where I started to look at protein folding and misfolding. So I was studying these calcium channels. For those of you who've taken cell biology, hopefully you're familiar with IP3 receptors, which form these channels in the endoplasmic reticulum membrane. When they're chronically activated, they're degraded by the proteasome through this process called endoplasmic reticulum associated degradation, or ERAD. I became fascinated with that topic. I did a lot of biochemistry and some electron microscopy of looking at this new complex of proteins that I identified as a PhD student. But I wanted to continue in the ERAD pathway. I saw how is it relevant to many diseases, including proteopathies, genetic and inherited proteopathies like cystic fibrosis, as well as neurodegenerative diseases. So as a postdoc, I actually worked on 2 different projects for a while, and then finally landed on the Drosophila project I just told you about. My first project was looking at the role of sugar modifications or glycan modifications on specific proteins in the ERAD pathway that lead to degradation of proteins such as the cystic fibrosis transmembrane conductance regulator, which is the gene that's mutated in cystic fibrosis. Eventually, I moved entirely into the Drosophila model, and all the while I was also developing my own F1. So this is Molly, Brian, and Sophie during my postdoc, demonstrated as fruit flies here, represented as fruit fly pupa here. But, these are old images, they're not much older. But with that, we will take any questions.
I was interested in how the wild type was able to bind with the mutant for the continents. I was curious if other proteins also aggregate with those.
With Huntington?
Yeah, or for any of the has anyone done any like mass spec on any of those?
Yeah, they're really hard to break apart because they're so stable, but the core is thought to be Huntington, almost exclusively.
Okay.
Around that, you see things like ubiquitin and autophagy components, and anything that it's usually thought that it's trying to degrade the aggregate, but gets caught up in the aggregate. So it gets sequestered into these very insoluble aggregates. So P62, lots of autophagy pathway components, proteasome components are found in those aggregates. But when you strip all that away around the core, it's really Huntington. Yeah, those other proteins really can't assemble into the fibrils because they don't have the right sequence.
Yeah, I'm kind of curious if like some of the different types of diseases can like cross-contaminate the proteins or whatever.
Yeah, so as soon as we have these tau flies, we want to see if we can get that
Yeah.
happening between Huntington and tau. And there is evidence for that, but whether it's within the same fibril core or not is a different question. So the resolution we have on the confocal is not going to tell us that, but the mass spec would be one that you have to use nasty chemicals, like formic acid, to break these apart.
It's just a follow-up to that. If you express something that was like a wild type Huntington, but was also fused like ubiquitin protein, or if you just like flooded a cell with a protein, do you think that would help break apart some of the fibrils, or because it does bind to the mutant version?
Yeah, I don't think that would probably enhance transmission.
Yeah.
There's some evidence that wild type Huntington actually enhance transmission because it has that poly-Q stretch. Disaggregases could help break them apart. So there's a lot of work in yeast looking at disaggregases, so enzymes that are responsible for breaking down these types of aggregates, which are more functional in yeast than they are in humans. Yeah.
So I know insects have kind of secondary brains almost depending on if they're especially strong flyers, they'll have thoracic ganglia. Are there protein builds up in these secondary ganglia?
We've never looked. Or even thought to look. That's a good point. Yeah, we've tried to do some work. Huntington's disease patients tend to develop cardiac defects. So we started expressing mutant Huntington in the heart of the fruit fly. We never really saw anything too exciting happen, but the interactions between the periphery and the central nervous system, whether it's other neurons or other cell types, would be really interesting.
Thank you.
Mhm.
Just because I want to understand better, did you say, so did, when RAB7, so that gene was knocked down, right?
We did knock down RAB7, but we didn't see an effect.
Okay, so which one did you knock down, and which one was knocked down that decreased the transmission of the mutant Huntington?
RAB10.
It was RAB10. So are there some like, I don't know, some cells that don't have like RAB10?
Yeah, from what I understand, it's ubiquitously expressed, so it's expressed widely. It does seem to be enriched in neurons.
Right.
We're looking at it in glial cells. It's expressed in glial cells, but not as high as in neurons.
So is there a way for you to kind of like, I guess, enhance that in glial cells?
Yeah.
So that it wouldn't-
Yeah, so we can use the same transgenic tools that I described for Huntington and overexpress RAB10, which is what Dawn is doing. One of the things Dawn is doing. Yeah, good question. There was another hand.
All right, I just curious, like if there are any clues or evidence like from lipid metabolites like ApoE or like pathways like could be linked to the like the AP.
Yeah, so I showed that that pedigree with the RAB10 Alzheimer's resilience locus. So those patients have the ApoE4 allele, so they have a much higher risk of developing late onset Alzheimer's disease, but for some reason they're resilient. The interaction between ApoE and RAB10 has never been, other than what I just described, has never been studied, to my knowledge. But it's a good question because membrane localization definitely enhances aggregation of all of these proteins.
switch from pre-med to like say not the struggle for you?
Yeah, I mean, I got into medical school, I think I said that, but so that was difficult to say no to. First I accepted, and then I said, never mind. Yeah, it was a struggle because it had been a lifelong dream, and there's a lot of factors involved in that decision, economical and personal and all that, but I knew that I loved being in the lab, and I don't regret it.
All right.
Any insights that you can share with students who are deciding, because a lot of our students are thinking medical
Yeah,
and then switching.
and get experience. Getting experience, get into labs, do multiple lab experiences and make sure you really like the discovery part of it. And on the other side, clinical experience, and then you have to get now, but back when I was a student, it was kind of optional.
Good suggestions for when students should start doing research projects.
As soon as possible. I tend to find after your first year, get used to college. That tends to be a really good time to start. Every student's different though. So I've had people start in my lab as freshmen. And they've done wonderfully. So sooner than later.
Are you accepting students?
Currently, we're maxed out. We're at capacity, but if people want to email me, I'm happy to say, contact me at this point to see if that's still the case, which I have done a lot recently, but I'm happy to keep communication lines open.
Thank you very much. Let's thank our speaker once more.